The overall goal of this procedure is to provide a convenient synthetic access to guanidine functionalized peptides.
This method can help answer key questions in the field of medicinal peptide chemistry, such as the development of integrin ligands or GPCR ligands.
The main advantage of this technique is that it allows an easy synthetic access to functionalized guanidines, completely compatible with SPPS.
The technology will be especially valuable for drug-like molecules, because the functional groups which are modified are typically decorating biomolecules. In our case, we aim for molecules which can be used for molecular imaging of distinct properties of individual cancers and for their specific treatment, an important issue for personalized medicine.
To begin this procedure, add 1 gram of Bach-pyrazole carboxamidine and 1 gram of PPH3 to a 50 ml round bottom flask. Subsequently, add 10 ml of dry THF to a 50 ml round bottom flask in an inert atmosphere via a rubber septum. Using a syringe, add 194 microliters of dry methanol. Stir the solution at room temperature. Using a syringe, add a prepared solution of diisopropyl azatic in THF dropwise over 15 minutes. Then, let the solution stir for 2 hours at room temperature. Using a rotary evaporator, remove the solvent under reduced pressure. After this, dissolve the crude product in 1 ml of DCM.